All right, welcome back. We're going to keep talking about cells. In the last video that you watched, we talked about the function of the different organelles in a eukaryotic cell. And now we're going to get into the actual life cycle. What is the cell doing at different points? How does it split? That sort of thing. And then we'll end up with a conversation on what cancer is and maybe a little bit about why it's bad. Now, I will say we're not going to go into as great a detail about the cell cycle as you may have in your biology class. We just want to dig up some old memories and do a quick overview before we move into some of the other stuff. So, looking at this, the life cycle is basically all the different things that the cell goes through and approximately how long that it goes through. And so a big part of this, ultimately, when we talk about the life cycle, we talk about cell division, right? And then we get into that horrible M word, mitosis. Where meiosis is a special kind. We'll talk about that at the end of the school year when we do reproduction. So cellular division is a type of cellular reproduction where a single cell divides into two daughter cells. Now, those daughter cells are genetically identical, and that's going to be important for, if you have skin cells, you want them to all be the same skin cells or heart cells or muscle cells, that sort of thing. At the end of a cell's lifespan, which is programmed, there's a certain number of divisions, basically, that a cell goes through before it dies, basically. And then it goes through genetically controlled death, also known as apoptosis. So you reach a certain number of copies, and the cell knows that it needs to destroy itself. And that's just to protect the rest of the body. If a cell gets too old, there could be some genetic damage that could carry on. And aging is actually, one way to define it, is the accumulation of genetic damage over time. And so to try to minimize that, cells will program themselves. And there's also the cancer element also, which we'll talk about, like I said, at the end. And so here's an overview of what the cell cycle is all about. And so basically you can divide up the life cycle of a cell into different phases based on what the cell is doing. So some of the first ones we look at, interphase, can be divided up into a few different sections, G1, S, and G2. The actual cellular division part is this mitosis section right here, including cytokinesis. Now, it's outside of that wedge because it's part of it, but it's not a specific step or stage in mitosis. And then sort of things start over here at this G0 before the circle continues again. So we're going to dig in a little bit to these various cycles, and we're going to start with G0. And so at this point, the cell is not actively preparing for division. So either A, division has already happened, or B, division is not going to happen. So this phase, you've got, you can kind of think of it as like regular cell life, okay? The cell is just doing its normal day-to-day thing. So some example, like your mature cells, your skeletal cells, and your neurons, once they reach this stage, they never leave it. That also means that they're not going to divide, okay? Other cells that are going to divide, skin cells, stem cells, are going to maybe skip this step, or they may spend just a very brief time before they go back into the preparation for the actual division. Then we have G1. This is the first phase where the cell is still doing normal function, but we are, the cell is growing. We're building new organelles. We're increasing protein synthesis to get ready for division. This is sort of the gear up phase, okay? We start to get a little bit of movement of the different organelles as they're moving around and stuff like that. This can take as little as 8 hours or up to, if it takes several weeks, we've probably got some sort of cell division that's about to take place. Moving into this, in the middle of the two G phases, G1 and G2, we've got the S phase. Now we call S is for, let me get a different color. S is for synthesis. Specifically, we're talking about DNA, and this is where the DNA copying takes place, and DNA replication with the helicase and the DNA polymerase and the matching the base pairs and all that other stuff that you talked about in biology. We're not really going to go into that. You just need to know that DNA replication is happening here. Okay. Here's a quick overview if you want to pause the video real quick and sort of take a look at the different steps of DNA. We've got the splitting of the DNA helix right here. Remember, DNA is a double helix, that sort of spiral staircase looking thing. We've got DNA polymerase building new DNA strands off of the old ones. This is where we get into leading and lagging strands, which you may have mentioned when you were a freshman. And that has to do with, how we fix holes and stuff like that, like right here with your lagging strands. Then they get finalized and so on and so forth. So if you want to take a moment, pause it. You can look at this in a little bit greater detail. If not, I'm going to go ahead and move on to the other phases then. So then after DNA synthesis, we have G2, and so this is really hardcore protein synthesis. Now, protein synthesis, you also talked about it a lot in biology, right? Protein synthesis has the transcription and the translation, and we've got the tRNAs and codons and stop codons and start codons and all of this other stuff. Then you start getting into the mutations and stuff like that, and missense and nonsense and insertion and deletion, all that other stuff, okay? We're not going to talk about any of that either. What you just need to know that at this point, the DNA has already been replicated. We're doing last-minute protein synthesis and getting the centrioles copied so we can get ready for mitosis. Now, the G1 and G2, those serve as checkpoints, right? And so the cell is not going to move out of G2 into cellular division until we've passed through this phase. So this gives the cell a chance to do any last-minute changes before it has to move on to the actual division part, which is the mitosis. That's the one that everybody talks about. Now, the majority of mitosis, which can take anywhere from 1 to 3 hours, is the movement of the DNA, right? That's what we're talking about. Cytokinesis is the last bit. That's the physical separating of the cell. Most of what we want to be talking about with mitosis is what are we doing with the DNA? And we've got PMAT, right? Those are our different phases: prophase, metaphase, anaphase, and then telophase. So we're going to spend a little bit of time talking about those. And just remember, the rest of the cell, all those other organelles, those are going to split too, but we're going to spend our time talking about what happens to the DNA to get those genetically identical daughter cells, okay? So here's a picture of an actively dividing cell before we get into the actual division and stuff like that. And what you can actually see here, this is what cytokinesis looks like, this sort of dimpling of the cell, and then eventually it just sort of pinches off and then they separate. Kind of like if you take one of those long balloons and then you twist it so it looks like you have two separate balloons, but they're separated by a little nubbin of rubber or whatever in there. Now pretend you pinch that off all the way and seal the two balloons so they're separate. And that's basically what ends up happening in that last stage. So let's look at these real quick. If you downloaded the notes slides, you have already drawn images. I'm just going to kind of draw these in. So if you'd like to draw your own, you can draw along with me. So early prophase, so prophase is basically the the DNA sort of collecting, or I guess you could think of it as solidifying. Basically, when you look at a nucleus, it kind of looks like a sort of cloudy ball, because all that DNA is just the individual strands floating around and stuff like that. In prophase, what we end up doing is gathering up all of that DNA, so it's easier to separate. So if you're looking at a picture of a cell, so here's our cell, here's our nucleus, okay? It's not to scale, okay? But basically what we end up having going on is our DNA is starting to come together to make the chromosomes that we're used to seeing, right? Your centrioles, think of those as kind of like pulleys or winches, okay? Those are moving to either side of the nucleus, to the poles of the nucleus. Those are the things that are going to be doing the actual pulling. The other thing that's starting to happen during prophase is the nuclear envelope. So this thing right here is going to start dissolving in order for the DNA to start to separate. And so by the time you get to the end of prophase, late prophase, what you're going to end up with is you've got your cell with the nuclear envelope more or less gone. Your centrioles are going to be at either end, right here and right here. And then you're going to have spindle fibers are going to start to attach to your chromosomes. So they'll be kind of like this. And then your chromosomes are here like this, okay? Now they're not going to be completely lined up yet. They're just going to be attached and they're going to start lining up. They don't actually line up to be separated until you get to metaphase. So here's metaphase. I've got my centrioles here and here. And I have my spindle fibers attached to my DNA. And so now they are aligned along the middle of the cell. They're getting ready to be pulled apart. All right. Then we get into anaphase. Anaphase is when we pull apart. I'm going to shorten this for the sake of time, so we're just going to pretend that there's only one chromosome in here. My individual chromatids, draw that a little bit better. My individual chromatids are going to start separating, okay? That's going to give us our, ultimately, our two daughter cells. Now, in telophase, the chromosomes have separated, okay, into two identical chromatids, right? And then we're going to start to have the separation of the two cells and the reformation of the nuclear envelope, okay? The other thing that's going to be happening is your DNA is going to kind of start disappearing as it unwinds itself in these newly forming nuclei. That's basically the end of mitosis with those slides right here. Cytokinesis is when we completely pinch off the cells. Here's our nuclear envelopes. The centrioles, the centrioles are going to kind of go back to where they were or dissolve. The spindle fibers are going to go away also. And then we have two completely identical cells when there used to be one, and then we get into that G0 phase, okay? Okay, so that's basically the cell cycle, right? A couple of things, the mitotic rate, that's basically how fast mitosis is taking place, and therefore how fast Cellular division is taking place. A slower mitotic rate, so a lower number of divisions, means a longer cell life, because you're going to reach that maximum number of divisions less quickly. Because remember, programmed cell death is determined based on the number of divisions. So the faster you divide, the more quickly you're going to hit that. And so the shorter your cellular lifespan is. This process requires energy to do. Now, we didn't go into where the energy is used, like in the centrioles, for example, it's used there, but you just need to know that this is an energy intensive process. A couple of exemptions, I guess, to this. That cells that don't really divide, muscle cells and neurons don't divide. You've probably heard a lot that you're born with all the brain cells that you've got, and you're never going to get any more, and so that's why they tell you why you shouldn't use drugs and stuff like that. Other cells will divide more frequently. For example, skin cells and cells in your digestive tract, like in your stomach and stuff like that, will only live a few days or a few hours, and then are replenished quite frequently by stem cells, and then through genetic expression, those stem cells turn into skin cells or digestive cells or something like that. Other cells are kind of in between those, where they can live for several days or even weeks because they don't have to divide that frequently. Connective tissues, for example, would be one of them that needs to be replenished, but not as quickly as like skin cells, for example. Regulation of the cell cycle and cell division. Typically, you're trying to balance off cells that you lose by regrowing new ones. You don't want to have extra cells unless you're actively growing. Once you reach a certain point, when you stop growing, your cell division, your mitotic rate tends to go down. If it's going up when you're not growing, that could be a sign of cancer, which is a bad thing. Cell division can be stimulated by internal factors or extracellular chemical factors, like growth factors, certain factors that are released at the site of an injury because of inflammation or something like that, that can trigger cell growth and things like that. It can be inhibited by repressor genes that stop the cell division based on certain chemical cues that the cell is receptive to during G1 or G2, in between that synthesis phase. It can also be inhibited by telomeres. That's basically what determines when a cell is supposed to die. It's basically an end point, and each time the cell divides, that telomere gets shorter and shorter and shorter. Once it disappears, then the cell can no longer replicate, and then you go through apoptosis. All right, so this is when things are supposed to be happening normally. When they don't happen normally, you get into cancer. So a tumor, also known as a neoplasm, is a mass that's produced by abnormal cell growth and cell division. Now, not all tumors are bad. A not deadly tumor, I guess you could say, is known as a benign tumor. It's contained, and it's not life-threatening unless it's really, really large. Examples of this could be like moles on your skin are benign tumors, or if you have benign tumors in, they could happen in, let's see, where can they happen? Lungs, ovaries, places like that. Your doctor would probably keep an eye on them in case they turn threatening, but they won't do anything about them. Moles are another example of that one, where they will do something about it if they have to. However, if it continues to spread or becomes life-threatening, or when it really becomes a bad problem is when it spreads to surrounding tissue. That's known as invasion. That's also called metastasis. If it metastasizes, it's spreading, so parts of the tumor are breaking off and then growing in other places. when it becomes malignant, then it becomes cancer. There's an abnormal proliferation of cells. Typically that's caused by a mutation in the genes involved in cell growth. So remember those repressor genes that we talked about? If those are damaged in some way or they're not read properly, you can get uncontrolled cell growth. You can have, these genes are called oncogenes. Let's see, things that can cause this, right? So there are chemicals called mutagens that directly mutate, change the DNA that can lead to this. Carcinogens. Carcinogen is cancer-causing substance. A lot of them are mutagens. Nicotine, for example. A lot of compounds in cigarette smoke are known carcinogens that can lead to the generation of these faulty genes and then leading to cancer. Metastasis, I kind of jumped ahead, I apologize. It's the spread of cancer to other areas. That begins with invasion of the tissues of the surrounding tumor, especially if it gets to a spot where it can get into like the bloodstream or the lymph, which are major transport passages. Then you can get into metastasis that way, and then you can get into different stages like stage 1, 2, 3, and 4 and stuff like that. Here's kind of what that looks like, basically. So you've got your abnormal cell right here, and then that abnormal cell divides. And a lot of times these also divide very rapidly as well. And so eventually, if you look, after a number of cell divisions, you have cells right here, right, that are breaking off, and now these cells are going into the bloodstream, and so they're floating around, and then eventually they'll reach another spot someplace else. They'll get out of the blood vessels and penetrate the surrounding tissues in a new part of the body, and then start dividing again there. And so now we have a secondary growth someplace else, and this is really, really bad because it becomes really hard to treat this cancer if it keeps spreading around. You have to constantly keep looking for it, and it's really, really difficult to make it go away completely. So, okay, so I had talked about cellular differentiation. That's sort of branching off of the cancer thing a little bit. So cells start out as stem cells. They're blank slates, okay? It's a blank piece of paper, right? All of the cells contain the same chromosomes and genes. What happens is, in order for a cell to become a skin cell or a muscle cell or an eyeball cell or a small intestine cell or something like that, certain genes are turned off or on based on what the cell is going to turn into. Okay, so certain genes are turned off, and so then that cell becomes a muscle cell or something like that. Liver cells, fat cells, neurons, and stuff like that. And when we get into those specific tissues, we're going to look at, like, nerve cells are highly, highly specialized cells. Fat cells, goblet cells in your stomach are very specialized cells that have to act in a very specialized way. And so those genes are going to be activated or deactivated in order to create that cell. It's got all the same chromosomes as like a liver cell, but they're going to behave very, very differently. And that's because of how we read them. And that's basically it as far as the cell cycle and all the cell growth and stuff is concerned. Hopefully a lot faster than you've probably studied in the past. I'll have some models up that you can view that looks at the different stages of mitosis in the cell cycle if you'd like to take a look at those. They'll be up in the classroom, and then I can put it on the live feed for the folks at home. I'll set them up on that back table there. Make sure you go through and do the discussion elements. There's a little bit of some stuff about cancer, why it's important, a little bit more, so make sure you get those done. If you have any questions, send me a message on Remind or email, hit me up on Zoom or see me in class or something like that. Thanks for watching, and I look forward to seeing you all again.